Hi, I'm Adam Steele, a postdoc in Caroline Robertson's lab at Dartmouth College. Thanks for coming to my talk, The Place Memory Network, a network of brain areas supporting perception and memory of familiar places. Thank you very much to the organizers, and thank you all for being here. From the moment this image appears on the screen, you begin performing visual analysis. You can, for example, identify that this is a street where you might walk or drive here, and you see some trees and objects. All of this happens as soon as the picture appears before you. But the ability we have to do scene recognition is not reflective of how we generally experience the world. More often, when we encounter a visual scene like this, we have brought ourselves here. We have navigated to this place with a purpose. In this case, to go to Mama Llama's eatery and cafe. So, for the brain, the goal of scene processing is not necessarily scene recognition. More often, the goal of scene processing is to service navigation. And navigation requires knowing what we want to do and what we will see in our environment along the way. For this reason, we would expect that the brain areas involved in scene perception should closely interface with the memory areas to provide context for the current field of view to support memory-guided behaviors like navigation. A lot is known about how the brain accomplishes visual analysis of scenes and how the brain represents spatial information. We know that there are 3 scene-selective regions of the brain, one on each of the ventral, lateral, and medial surfaces, and these areas collectively identify landmarks, determine navigable spaces, and derive other types of useful visual information from a scene. We also know that the hippocampus and entorhinal cortex are key areas for place memory and contain specialized cells for representing spatial information such as place cells and grid cells. But less is known about how these two systems interact. Specifically, how is contextual information conveyed to the scene perception areas to shape ongoing perception in order to influence what we will attend to and where we will direct our gaze. 2 disparate lines of research have led to different hypotheses about where in the brain this interfacing might occur. First, memory and perception could share neural substrates. The evidence for this comes primarily from mental imagery, which is a type of explicit memory recall. Many fMRI studies have reported that mental imagery activates the same brain areas as perception, including low-level visual areas like V1 through high-level areas like category-selective ventral temporal cortex. In fact, the classic understanding is that the neural substrates of perception and memory are the same. Alternatively, memory and perception may have distinct neural substrates. The evidence for this is more circumstantial, and it comes primarily from analyses of resting state functional connectivity. These studies have found that the anterior PPA is more functionally connected to the anterior aspects of other scene-selective areas and to the hippocampus, suggesting that there may be a transition from perceptual to memory-based representations moving anteriorly along these areas. However, this has not been shown empirically outside of resting state functional connectivity data. In this talk, I will describe 3 experiments where we investigated these hypotheses. We began by asking whether perceptual areas support place memory recall. This question can be addressed by looking at functional topology. So we wanted to intensively map scene-selective perceptual activation and place memory activation in 14 individual subjects to enable a fine-grained comparison. We performed 2 different functional localizers. The first localizer was the scene perception localizer, and the second localizer looked for place memory preferring regions. For the scene perception localizer, participants saw blocks of scene, face, and object images presented rapidly, and we compared bold activity during scene versus face blocks. For the place memory localizer, participants provided a list of 36 personally familiar people and places. In the scanner, we showed participants these stimuli and asked them to recall the people and places as vividly as possible. As I said, we characterized the perception and memory localizers in individual participants. So I will be showing you individual participant data to give you a sense of how that looks. The group data will be presented in these rows plots. So circling back to the hypotheses that I introduced earlier, here you have a possible configuration of the perception localizer in white and the memory activation in burgundy. The center of mass of the perception cluster is the center of the rows plot. and the vector represents the center of mass of the memory cluster. The angle of the vector represents the angle between the two clusters, and the distance shows the separation. Here you can see that the clusters are primarily overlapping, and the vector is not very long, suggestive of the first hypothesis being true, that perception and memory share neural substrates. Alternatively, the perception and memory areas could be further apart from each other, resulting in a long vector. This would be consistent with the second hypothesis. Or you could have a different configuration altogether, and again, the Rose plot would reflect this. On to the data. Here is one subject's scene perception localizer. This is the ventral surface. In hot colors, you see the scene perception area PPA. In cool colors, you see the face selective area FFA. Here is OPA on the lateral surface and MPA on the medial surface. Now I will show you where the memory activation occurred. I'll focus first on the lateral surface. Again, this is scene selective OPA. Now I'm going to show you the memory activation. OPA will remain outlined in white. And here is the memory activation. As you can see, this is dramatically anterior to the perceptual activation, and there is only a small amount of overlap between these areas. I will flip back and forth so you can appreciate this difference. This anterior shift was present in every scene network area. Interestingly, if you take note of FFA here, you'll see that the face memory activation does not show this dramatic anterior shift. Here's a second subject's perception localizer, and here's their memory localizer. Again, the anterior shift of memory activation is really dramatic, and this is not present in the face areas. Now I will show you the Rose plots that summarize the group data. Anterior is going to be towards the top of the slide. The topology of responses was remarkably consistent. All subjects showed an anterior shift of place memory relative to scene perception on the lateral surface, on the medial surface, and on the ventral surface. And there was no significant shift of face memory responses in ventral temporal cortex, suggesting that this is a scene-specific effect. So do the scene perception areas support place memory? No, we find that they do not. In fact, we observed that each scene area has a paired memory area. We next asked whether the memory areas were functionally distinct. To address this question, we examined the correlation of the bold signal of these regions during naturalistic movie watching. Participants saw a concatenated series of college admissions videos and architectural tours, which had varying perspectives and viewpoints. Importantly, these videos all conveyed a strong sense of space. The whole movie was 11 minutes long. And we examined the correlation of activity between each ROI pair. And what we looked for was a greater correlation within each network, so perception to perception and memory to memory, compared to the correlation between these areas. Here you see the within network and between network correlations on the x-axis and R values on the y-axis. The within network correlation for both the scene perception and place memory networks was very strong, and the between network correlation was significantly lower. This confirmed that the perception and memory areas do segregate into distinct functional networks during naturalistic movie watching. We also looked at the correlation of activity between these networks with the primary visual cortex and the hippocampus to see whether they might be more closely aligned to the brain's perceptual or memory systems. And indeed, we found a double dissociation between these connections. The scene perception network was more connected to V1 than the place memory network, and the place memory network was more connected with the hippocampus. This suggests dissociable roles for these networks, and that the place memory network might bridge perceptual and memory systems in the brain. So yes, we did find evidence that the place memory network constitutes a distinct functional network from the scene perception network. Finally, we wanted to determine whether the place memory areas were involved in memory processes more generally and not just in top-down constructive processes like memory recall. So we asked whether the place memory areas showed a familiarity effect when participants saw recognizable stimuli. To investigate this question, we had participants view panning movies of familiar versus unfamiliar places. These videos were of locations that participants told us they were familiar with in the real world. The videos looked like this, sweeping from left to right and back again. And we asked participants to imagine that they were in that place, turning their heads from left to right. We compared the activation of the place memory and scene perception areas when participants were viewing movies of familiar versus unfamiliar places. The greater response to familiar videos than unfamiliar videos was the familiarity effect. We hypothesized that the place memory areas would show a greater familiarity effect than the scene perception areas. And indeed, that is what we found. This is data from each cortical surface. On the y-axis is the difference in activation when viewing familiar compared to unfamiliar places. On the lateral surface, as you can see, the place memory area showed a greater familiarity effect than the scene perception area. This was true on the ventral and also on the medial surface. So the answer to our third question is yes. These areas do preferentially activate for familiar visual stimuli, suggesting that they are involved in recognition memory in addition to memory recall. So, to summarize, I have shown you that distinct areas of the brain are activated to support place memory. These areas fluctuate as a network and respond preferentially to videos of familiar places. Each of these areas is paired with an area of the scene perception network, suggesting that they may be a crucial link between perception and memory in the brain, providing contextual information to inform ongoing perception during navigation. With that, I will thank my co-authors, Caroline Robertson, Madeline Billings, and Ed Silson, and the rest of the Robertson Lab. For more information, you can check out our preprint that just posted on bioRxiv, which has additional experiments as well. I'd also like you to check out Caroline Robertson's poster, which discusses these data in more detail. And with that, I'll take questions.